Hey everyone, and welcome. You ever just look at the world around you and wonder how we got all the different plants and animals we see every day? Well, for thousands of years, humans have been, kind of playing the role of genetic designer, intentionally picking and choosing the traits of other living things. So today, let's dig into how this whole practice of genetic design actually works.
I mean, just look at these dogs. We all know them. But they didn't just show up like this. We humans actually bred St. Bernards for that incredible sense of smell, Huskies for their sheer endurance to pull sleds forever, and German Shepherds for their amazing intelligence. We literally picked the traits we wanted and guided their evolution.
So that's the big question, right? How did we actually pull that off? How in the world do you go from, say, one common ancestor to this wild variety of specialized animals? Well, the answer is way older, and honestly, a lot simpler than you might think.
It all comes down to a process called selective breeding. And no, this isn't some brand new high-tech lab invention. It's something humans have been doing for ages. It's this simple but really powerful idea of choosing which organisms get to pass on their genes, which makes the traits we want more and more common over time.
And to get the results thereafter, breeders have really honed in on two main strategies, two really powerful tools in their genetic toolkit.
So on one side, you've got inbreeding. That's all about keeping traits pure by breeding organisms that are super closely related. And on the other side, you have hybridization, which is all about mixing it up, crossing different parents to hopefully create a brand new, awesome combination of traits. Let's start with hybridization, the art of the genetic mix and match.
All right, first up, hybridization. The best way to think about this is like you're trying to create a genetic greatest hits album, combining all the best traits from two different parents into one.
So what are breeders really trying to get? Well, things like making a plant resistant to disease or helping an animal grow faster. For example, a breeder might take a tomato plant that's really good at fighting off diseases and cross it with another tomato plant that grows super fast. The goal? A hybrid that has both of those amazing qualities.
Now, the advantages here are pretty obvious. You can get organisms that are tougher, more nutritious, and just better adapted to their environment. But, and this is a big but, it is not easy. The main downsides are that it can be incredibly expensive and take a ton of time. Get this, it took breeders 3 whole decades just to develop high-yield hybrid rice varieties. 30 years!
Okay, so that's mixing. What about the opposite approach? Our second strategy is inbreeding. If hybridization is all about creating new combinations, inbreeding is about keeping the traits you already have as pure as possible. And this really gets to the heart of it. Anytime you hear the term purebred, you're hearing the result of inbreeding. Think about Clydesdale horses. They were specifically inbred for that strong build and obedient nature that they needed for farm work. The whole process makes sure those specific traits are passed down generation after generation. The upside is you can get rid of traits you don't want and really lock in the ones you do. But there is a very serious risk here. Because you're breeding organisms that are so closely related, you dramatically increase the chances of passing on harmful hidden traits. If there's a dangerous gene hiding in the family's gene pool, like the ones that cause hip problems in some dog breeds, inbreeding is way more likely to bring it to the surface.
Okay, but this brings up a huge question, right? You can see an organism's traits on the outside. That's its phenotype. But you can't see its genetic code, its genotype. So how do you know what's actually hiding in the genes?
Well, luckily, they have a tool for exactly that. It's called a test cross. You can kind of think of it like a genetic paternity test. It's a really clever way to breed an organism just to reveal its hidden genetic secrets.
So how does this genetic detective work? It's basically a 3-step playbook. First, you take your mystery organism, the one showing the dominant trait you're curious about. Second, you cross it with an organism you already know for sure is homozygous recessive for that same trait. And third, you just sit back and look at the kids. The traits they show will tell you everything you need to know about the mystery parent's genes.
Let's make this real with an example. Imagine you're a grapefruit farmer. You know that white fruit is the dominant trait over red fruit. You've got this awesome tree that produces white grapefruit, but you need to know, is it genetically pure white, or is it secretly carrying a hidden gene for red fruit?
So, to find out our tree's secret, we do the test cross. We breed it with a red grapefruit tree, which we know has to have 2 recessive genes for color. These Punnett squares right here show the only 2 ways this can possibly play out.
And this is where the magic happens. If your mystery tree is homozygous dominant, meaning it's got 2 copies of that dominant white gene, then 100% of its offspring will have white fruit. Simple. But if it's heterozygous, with 1 white gene and 1 hidden red gene, then half its offspring will be white and the other half will be red.
And that 1-to-1 ratio is the smoking gun the breeder is looking for. The second you see even 1 red grapefruit pop up, boom, case closed. You know for a fact that your parent white grapefruit tree was heterozygous. The test cross has revealed the truth.
All right, so let's pull this all together real quick. Selective breeding is just humans choosing the traits they want. We do it through hybridization to mix and match different traits, or through inbreeding to keep traits pure. And when we need to peek behind the curtain at the genes, we use a test cross to reveal an organism's genotype.
I mean, when you think about it, this is a seriously powerful toolkit. It's shaped our food, our pets, and our entire world for thousands of years. Which leaves us with a really fascinating question. Now that we understand the rules of this genetic game so well, what comes next? What should we design?